Welcome to your chemistry online class. In last class, we have discussed about sweeter ion and vitamins. So in this class, we are going to discuss about the structure of nucleic acid and what are nucleic acid and what are the functions of nucleic acid, all those things. So, nucleic acids are first called as nuclein because it is identified from nuclei of pus cells. Then it has acidic property. So based on that, it is called as nucleic acid. So we'll see what are nucleic acid. It is a polymer. So it is made up of lot of monomer and consists of nucleotide residue. So the monomer units are nucleotide. It is located in the cell, in the nucleus of the cell. And our all hereditary properties, and we are similar to our parents, those all things, and either our nature, all those are similar to our parents. Such hereditary properties of all are determined by this nucleic acid, and it is mainly made up of its elementary composition, that is elements are in nucleic acid is carbon, hydrogen, oxygen, nitrogen as phosphorus. So nucleic acid are made up of mainly these elements. And there are mainly two types of nucleic acid, these are DNA and RNA. The full form of DNA is deoxy ribonucleic acid. RNA is ribonucleic acid. Then it is present in both eukaryotes and prokaryotes. And how it is distributed in eukaryotic. In prokaryotic it is RNA, in eukaryotic it is DNA. So it is presented in the form of, it is presented in nucleus with small amounts in mitochondria and chloroplast. So it is mainly found in nucleus, and nucleus consists of mitochondria and all those things. It's in those also it will present. And RNA is found in throughout cell. So nucleic acids, we have said that nucleic acids are polymer. So it is polynucleotide. The building blocks are nucleotides. What are nucleotides? These are energy rich compounds that helps for the, or that is the main part of metabolic process of cells, and it serves as chemical signals, key links in cellular system, and it responds to hormones and other extracellular stimuli. In we know our body having certain hormones, and each hormones having certain function, and each cells and each nucleic acids and the messages, the reflex action, nerves, all are controlled by these key links called nucleotide. It is a structural component of almost all enzyme cofactor, and it is a metabolic intermediate. And each nucleotide having three parts or three units, those are phosphate, sugar and nitrogenous base. We will see each one by one. So a nucleotide consists of a phosphate linkage, sugar, this sugar is two types, ribose or deoxyribose, and base are two types, mainly purines and pyrimidines. In purines, that are adenine and guanine, in bases that are cytosine, thymine and uracil. The bases in this RNA, there are RNA and DNA. So these bases in RNA and DNA are different. Only one difference is there that we'll see. So a phosphate linkage is there. That is H3PO4 contains 3 monovalent hydroxyl group and a divalent oxygen group. So there is phosphate linkage and sugar. Pentose sugar is there and ribose sugar is there. Ribose is there. Deoxy ribose is there. In deoxy ribose, only one difference between ribose and deoxy ribose. Instead of one OH group in ribose, there are only hydrogen. So there is an absence of oxygen. So it is ribose. And another one is deoxy ribose. And this pentose sugar form esters with phosphoric acid. The pentose sugar and phosphoric acid having a certain link. So this bond is known as phosphodiester bond of nucleic acid. And nitrogenous base is two types, purines and pyrimidine derivatives. And this is also so if we take DNA or RNA, there are certain links between phosphoric acid and glucose, link between base and sugar. Such links you can see. And if we take the bases, there are certain pairs of bases, that is these purines are adenine and guanine, pyrimidine derivatives are uracil, thymine and cytosine. They are also linked in a certain form in DNA that we'll see in the structure. So this total structure is called nucleoside. That is a link between sugar and base. There is a link between sugar and base. Such structure is known as nucleoside. That is when a ribose or deoxyribose is combined with purine or pyrimidine base gives nucleoside. So here we can see the ribose sugar and deoxyribose sugar. So only difference we can see here, OH is there, here H is there. This is the only difference between ribose sugar and deoxyribose sugar. So here you can see the phosphate and sugar backbone. So if you take the nucleic acid structure, you can see a phosphate and sugar linkage. So the nucleotides are all oriented in the same direction. So it is always joined the third carbon of one sugar to the fifth carbon of the next in line. For example, if you take, so this carbon of sugar will be third one, this one will be fifth one. So the first phosphate linkage with the third sugar and the second phosphate linkage with the fifth sugar. Like this manner it is arranged. So it is 3-5- model. Then it is added to the base. That is, so it is consist of one phosphate linkage, sugar and base. So there is also sequence in the arrangement of bases. If you see here GCCATT, like that way all bases are arranged. So the bases are arranged the first carbon, and its order is very important. If we take the DNA, for each genetic character having certain base code. So we'll detailedly see what is DNA. Every living organism contains DNA. It is the material of inheritance and it is discovered in 1960 by Franklin, Watson and Crick. And if you see DNA, it is having a double-stranded structure. This double-stranded structure proposed by Watson and Crick, and they got Nobel Prize for their invention of, that is, the structure of DNA. So it is very famous. For this, they conducted series of experiments and concluded that DNA is the genetic material present in the nucleus of cell. So, if you take human DNA, it contains 3 million deoxy ribonucleotide residues and contains 25,000 genes. And for our inheritance, these genes are carriers of our inheritance. So here you can see the exact structure of DNA. So it is a double-stranded structure. If you see here, guanine is always bonded through a triple bond. This bond is hydrogen bond. Hydrogen bond is there. So guanine having always a triple bond, and adenine and thymine having single bond. So always the order is ATGC. That is, adenine always connected to thymine, guanine is always connected to cytosine. It is triple bond, it is double bond. So this is ATGC. Always it will be like that. There won't be the combination between adenine and guanine or thymine and cytosine. It will be same. Adenine, thymine, guanine, cytosine. So this is the double-stranded structure of nucleotide, that is our nucleotide, that is polynucleotide, that is our DNA. So it is made up of 2 strands of polynucleotide. So these are anti-parallel to each other, that is, if you check here, you can see here it is 3 dash. It is started from 3 dash, but it is here it is started from 5 dash. That is, if you check here, it is attached to the 5th carbon. So this model is also known as 3 dash 5 dash model. So these all are joined by the bases. So I already said each base is paired with a specific partner. That is, adenine is always paired with thymine, guanine is always paired with cytosine. That is, each purine is with each pyrimide. Thus, the sister strands are complementary but not identical. These are joined by hydrogen bond. These are complementary to each other but they are not identical. And here you can see the structure of the purine bases and pyrimidines bases. These are purines, these are pyrimidines. And this is the Watson and Crick model of DNA. This is for this structure. These are Watson and Crick. For this structure, they got Nobel Prize. This structure is also known as double helix structure. So, structure of DNA. Primary structure, secondary structure and tertiary structure. Primary structure, it is made up of nucleotide residue. Secondary, it is pertains the helix form to DNA strands. Tertiary refers to 3-dimensional shape, that is showing our Swiss spread and comet shape. And DNA replication is a main property of DNA. That is, DNA in our body, DNA carries all information necessary to making a protein in living organism. So proteins is synthesized by this DNA. So all information how to form a protein, this all information carried by DNA. So this DNA store this information and it should pass to the new generation. That is, if my mother having all nature, that is my mother's DNA having certain natures, and it is stored how the proteins are synthesized in her body, and it will give to the new generation, that it's to me. So this all have to give. So this process is known as DNA replication, and this information is all carried by primary structure of DNA. So to pass information stored in DNA to a new generation of cells, DNA replication must take place. Then when DNA is replicated, each strand of double helix, it is form a new strand of DNA, that we can see the image, then you will be clear with this. So so a new strand will form, that is known as daughter DNA. So one strand from the parent DNA is present. You can see that. This is called as, if it form one strand, that is called this is called as semi-conservative replication. And the production of new DNA is carried out by enzymes called DNA polymerase. So these all are not much important for you. DNA polymerase catalyzes the addition of deoxy ribonucleotide residue as a growing DNA strand. Here you can see this. This is the parent DNA. The parent molecule has two complementary strands of DNA, each base is paired by hydrogen bond. Then the first step, it will separate and it will form as two strands. Here it is as one, it is separated as two strands, and each parental strand now serves as a template, and it will form with another strands, and each new strands formed here. These are daughter DNAs. This one is daughter DNA, this one also daughter DNA. This is the process of DNA replication. So here you can see the translation. Then we'll move to RNA. RNA is ribonucleic acid. It is also similar to DNA. It is a long unbranched macromolecule consists of nucleotides joined by 3-5- phosphodiester bonds similar to our DNA. The structure of RNA, its structure is similar to DNA. Both are sugar-phosphate polymers and both have nitrogen containing bases, that is we have seen in DNA both having so similar to this. It is also having phosphate and sugar bond, and nitrogen bases are attached to this. But mainly there are three differences between DNA and RNA. RNA contains a different monosaccharide residue, that is there ribose. Here there is deoxyribose sugar, but here ribose sugar. Then DNA contains the bases of adenine, guanine, cytosine, but RNA contain adenine, guanine, cytosine, but instead of thymine here uracil. That is another different. We know DNA is double stranded and its double helical structure, but RNA existed as single strand instead of double strand. Here you can see the structure of RNA. Here you can see the bases in RNA. And there are different types of RNAs, mRNA, tRNA, rRNA, etc. So messenger RNA, transfer RNA, and ribosomal RNA. These are very important for you. Messenger RNA transfer genetic information from genes to ribosome to synthesize proteins, and transfer RNA transfer amino acid to messenger RNA for protein synthesis. And ribosomal RNA provides structural framework for ribosome. So the main function of RNA is protein synthesis. So for this, messenger RNA gives or transfer the genetic information from genes to ribosomes, and ribosomal RNA, that is rRNA, provides structural framework for ribosomes, and transfer RNA transfer amino acid to mRNA for the protein synthesis. So there is a co-work. So what is the protein synthesis? We will see the main process of this DNA and main work or function of DNA and RNA is protein synthesis. So these are the following three processes are involved in the duplication and transfer and use of genetic information. How the genetic information were transferred? The first step is replication. That is the process by which a replica or identical copy of DNA is made when a cell divides. That we have seen the replication of DNA. A daughter DNA is forming. And transcription. The second step is transcription. That is genetic message contained in DNA are read and copied. So first a daughter DNA will form and it will copy all the information, and translation. That is the process by the genetic messages carried by RNA are decoded and used to build proteins. That is from replication a daughter DNA will form that will copy all the messages, and these copied messages were decoded by RNA. And the proteins will form. So it is the first step is from DNA, it will undergo replication and then transcription, and it will form to, it will give the messages to RNA, then translation, it will form protein. This is the process. So transcription, it is the first step in using the information stored in DNA to produce proteins in transcription, using DNA as a template to make RNA. So from DNA, RNA will form. DNA is the main structure, from that RNA will form. From RNA, our protein will form. Several different types of RNA will form: mRNA, tRNA, rRNA. It is also involved 4 steps: initiation, elongation, termination and processing. So here you can see the steps, how it is forming. From DNA to RNA. So translation, that is the protein synthesis occurs in ribosomes, which are outside the nucleus and within the cytoplasm of cells. We know cell consists of nucleus and cytoplasm, mitochondria and all. And the mRNA connects with the ribosome, and the amino acids attached to transfer RNA are delivered one by one. And protein synthesis or translation takes place in 3 steps: initiation, elongation and termination. In initiation, a ribosome and mRNA and tRNA come together to form a complex. The elongation, amino acids are joined to the growing polypeptide chain. Then termination, the protein has been synthesized and the ribosome, mRNA, tRNA complexes dissociate, and they will further become mRNA and tRNA and rRNA, etc. Here you can see the process. First a complex form. So first step is they will come together, ribosome, mRNA and tRNA. Then elongation. First they will make a complex, then elongation, that is we know polypeptide chain. How a polypeptide chain will form with amino acid, that we have discussed. And termination, the complex will dissociate. So each step you can see, that is ribosome. Here you can see ribosome, rRNA, mRNA joined together, then give a complex. Then complex with amino acids. You can see here. Then termination. So all genetic messages or genetic nature is given by this RNA and DNA. These all things are there for you. These are the important features of DNA and RNA.